Can you hear me now? Hello? Hello?
Sir, am I audible, sir?
Yeah, you are audible, but how about me?
No, sir. Your voice is not coming, sir.
Why so?
No, voice is not coming, sir.
Now? Uh, Hello? Hello?
Hello, sir.
Can you hear me?
Yes, sir, you are audible, sir.
Am I audible?
Yes, sir.
Okay. There's some issues with the system this side. Can you, how about now?
Sir, now it is audible, sir. Actually, there is a fault in my system only.
Yeah, that's what I was trying to look into.
Now it is clearly audible, sir. Thank you, sir.
Okay, then fine. Okay, so let me just try with sharing things also once.
We'll start in a while, sir. Some participants are yet to join.
Okay. Um, are you able to see me?
Yes, sir. It's a shall we start or what?
Sir, excuse me, sir, your face is not visible properly. If you can adjust your camera.
No, this is a default camera. Okay.
Okay, sir.
I'll do that. First, I would like to share my presentation file.
Sir,
Sorry.
we shall give a formal introduction, sir. Then we shall start with the proceedings.
Let me first, set the presentation file.
Okay, sir. Sure.
Do you have an idea how we share here in Zoom? Because I generally don't use Zoom. I use the other systems.
You will be share. You can share your screen. There will be an option in the Zoom platform, sir. Below for sharing your presentation. So you can go to the taskbar, sir.
Yeah, that is the green button, no? Share.
Yes, sir. Yeah.
So when I just do that, screen sharing, I had to do it. Am I right?
Yes, sir. Yes, sir.
Yeah. Okay.
Okay. Shall we start, sir?
No. Wait a minute. I had to bring the file.
Sir,
Just a minute. I think I'm visible now, I guess.
yes, sir. You are clearly visible.
Wait.
So shall we should start with the formal introduction, sir?
Yes, please go ahead.
Okay. Thank you very much, sir. So welcome, dear minds, to this radiant new day where science and spirit together hold away. On the fourth dawn of our digital quest, we gather again, inspired and blessed. Good afternoon, all, esteemed speakers, distinguished delegates, respected faculty members, and enthusiastic participants. It is with great pleasure and honor that I welcome you all to the fourth day of our national webinar on digital frontiers in pharmaceutical research and innovation. Over the past 3 days, we have witnessed an enriching exchange of ideas, groundbreaking insights, and thought provoking discussions that have illuminated the evolving landscape of pharmaceutical sciences on this digital age. Let us approach today's session with curiosity, openness, and shared commitment to the advancing pharmaceutical research for the betterment of global health. It is my distinct honor and privilege to introduce our revered speaker for this session, Professor Dr. B.R. V.R. Padireddy sir, a distinguished academic and researcher from the School of Basic Sciences at IIT Bhubaneswar. Professor Padireddy's work delves into some of the most fascinating and foundational areas of modern chemistry. His primary research interests lie in the intricate fields of supramolecular chemistry, molecular recognition, and organic solid state and materials chemistry. His own academic journey is built on a strong foundation, having earned his PhD in chemistry from the prestigious University of Hyderabad in the year 1993. Throughout this illustrious career, Professor Peddireddy has been recognized with numerous accolades for his significant contributions to the sciences. He is a fellow of both the Royal Society of Chemistry and the Maharashtra Academy of Sciences, a testament to his international standing. He has been honored with the B.M. Birla Science Prize in Chemical Sciences, the Professor R.C. Shah Award from the Indian Science Congress, and was named as the best scientist of the National Chemical Laboratory. In addition to his research, he has contributed to the academic community as a co editor for the journal Acta Crystallographica, Section E. Today, Professor Peddireddy will be delivering a talk on a topic of profound importance in the modern drug formulation, pharmaceutical co-crystals towards enhanced bioproperties of the drugs. This presentation will shed light on how co-crystallization strategies can be employed to improve the physicochemical properties of the drug molecules, thereby enhancing their solubility, stability, and bioavailability. May I now request all of you for extending a warm welcome to Professor V.R. Paddireddy. Now, the stage is all yours, sirs. We are awaiting for your presentation.
Okay. Thank you for a kind introduction. I hope all of you are able to hear me. Hello.
Sir, yes, sir. You're quite audible, sir.
Okay. Thank you once again. Firstly, let me thank the management of C.V. Raman Global Institute and also the Dean Pharmacy, Professor Prushti, for inviting me to present the topic of interest from your side and also of my own interest on the pharmaceutical co-crystal towards enhanced bioproperties of drugs. I would like to take some time on this one to introduce the concept and also how these old concerns are being addressed through the new developments that happened in the last couple of decades. When I'm speaking at the Pharmaceutical Importance Institute or the department, I also have to bring some basic fundamental knowledge about this new concept because the experts and then the learners from this department, their orientation is been towards mostly different from the chemical concept and its application to the pharmacy. Because you all must be having very strong, solid founding in the bio evaluations of the drugs and also how to understand their interactions in vivo, either by experimental or by computational methods. But here, what I'm going to present you a new concept that has been going to be helpful, minimizing the a complex matter about the interaction of the biological system with the drug molecules. One of the important factors that I'm going to bring to your attention that we have been known for the accumulating lot of organic compounds and which have been used ultimately in the form of their superiority has been increased because some of them could be utilized as drugs to cure some diseases. In the past, maybe 30 years ago, I mean 3 decades ago or so, the number of diseases are also may not be of that high, and a few molecules that used to serve for the purpose. And as human beings, they start increasing their knowledge and application oriented mind to develop new drugs, then new diseases also started appearing, as I just believe it. And to address these new diseases, unlike in the past, whenever some new drug has to be introduced, then there will be serious complex procedures are being brought into effect and force due to the increased sense of ecology and human perceptions, and also not just limited to the humans but also to the overall animal kingdom. So that's why people are not that at ease, unlike in the past, to test the obtained molecules towards its drug activity by directly injecting to the human animal kingdom, which generally used to be considered the rabbits or the later on monkeys and so on, finally to the human beings. But in this kind of situations, how one can actually go for development of a new drug to cure the diseases of unknown nature in the past, and having the tremendous new phenomenon, new type of activity. Especially, we all had seen a few years back the very much complex situation about in the 2009 to 2020 era, that is in the COVID pandemic. It was really wonder, okay, because of the necessity and an emergency, though these tests are been off and then they made the vaccine to be available quickly, but in general that is not the case. Any new we haven't seen anything. That's why I'm sure all of you would agree with me, because you are all been in the mainframe process of this pharmaceutical aspects. I don't think that there is any drug that has been released into the market, a fresh drug, new drug, totally of a different concept, evolved from different concepts in the last 2 decades at least. The reason has been mainly the regulatory authorities to impose lot of constraints and conditions before releasing it. And due to this, it takes at least nowadays minimum 10 years to 15 years to introduce a new drug into the market. As a result, you can understand that. Yes,
ମାନେ
please.
mail କୁ mail ଆସିବ ନା ତତେ ଆଜି
Hello. Any concern?
No, sir. Please, sir. Carry on, sir. Please.
Yeah, somebody crossed up. Okay. So it is taking about 10 to 15 years, and naturally you can understand that when there are people, those who would like to just market it and then invest money and then market and get the returns, then it is unprecedented waiting period to get the returns on the value that they invested. Naturally, business is being business at the end, apart from the scientific intellectuality. So there are always a quest for new ways of just looking at the systems, how one can actually introduce drugs into the market, even if it is been a drug is been known already. There is another problem. What is that another problem? Suppose there is something that is being called generic drugs for the same disease, for known disease, and a drug works, and also there will be competition between from the generic people, and they have to wait. Manufacturers of the generic drugs, they have to wait until the patent has been expired. Once the patent expires, then people may not be able to really gaze it, or pre-evaluation can be done whether what will be the effect after the patent, because the innovator itself can also come with a new drug before the generic ones come into the picture. So these are all the things that has taken into account, and while people they started searching for the new avenues, how to make a drug that is been in a generic form itself, with instead of going for the totally a new drug brought into the picture. So as you all know that when generic drugs are being made, especially particularly with the same API, but either manufacturing difference or the placebo materials that are being used, and that makes a very difference for these generic drugs. But when the generic drugs that way just been prepared, there is always a problem for the bioefficacy or the bioproperty of the drug, because the placebo materials that are being used, that mostly inorganic materials, they create a big trouble in their effective activity of the drug that is an organic molecule to reach to the target, without which naturally the process cannot be made into a tablet because of the soft nature of the organic molecules. So naturally, one would see that bioavailability, that has been solubility, has been one major problem in this kind of generic drugs are being made. So there is a lot of thought process how to make get away from this complex problem using the any other techniques or other allied branch of the chemical sciences, allied chemical frontier concepts in the chemical sciences. So in that process, what I'm going to speak to you is evolution based on the co-crystals. That what I'll describe with you, I'll describe what is meant by co-crystals to develop some different type of pharmaceutical materials, and which shows different bioproperties. So this I'm going to explain in the next few minutes with some of the examples and some of the procedures that have been employed, and also how to characterize them. These are the concepts I would like to bring to your attention. I hope that you will have a take-home message to look into them from different angle, both from the academic research and also possibly to apply with your enhanced knowledge, and also the knowledge that you all been associated already with the pharmaceutics, with several drug molecules, to make them into reality by by bringing them to the bulk scale and probably towards the commercialization as well.
Sir,
Okay. Yes.
sir,
Yes,
sorry
sir.
to interrupt, sir.
Yes, please.
Sir, your slides are not visible, or
No, no,
your...
no. I have. No, slide is not at all visible.
No, sir, only the first slide, I
Yeah.
think it's visible, protein kinases.
I have moved slide at.
Okay,
Yeah.
okay,
Okay.
okay.
Yeah. When I'm just going to present you, whatever I given is the introduction so far of the talk. Okay, so let me take you now. Can you see the slide change now?
No, sir.
Huh?
Yes,
No,
it's
sir.
slide change, na?
Yes, yes, yes.
Now it shows drugs.
Yes.
Drops. Okay, sir. Slide changing.
Yeah, okay. So now if you just look at what I've been talking about. First, when you're talking about organic molecules, as I mentioned, they've just been used for some purposes of the human concerns. That has been nothing but the drugs. Then what is a drug? That we already discussed about the drug that has been made in the laboratory. You can see that one on the on my right side, the picture where the drug molecules can be prepared in the laboratories, and on the left side, the capsules that has been going to be in that form using placebo ingredients and having the active pharmaceutical ingredient that has been API present in the capsule that you see that on the my left side. Okay, these drugs, how you one qualify some molecule as a drug. Let us just look at from the pharmaceutical concept, then that is been based on the Lipinski's rule of 5. I'm sure all of you have been well aware of this one. Just to say that one, for a molecule has to be more likely to be absorbed and distributed effect, having the following 5 concepts are being fulfilled. One is that it should not have more than 5 hydrogen bond donors and not more than 10 hydrogen bond acceptors, and molecular weight, it should be at least less than 500 Daltons, and then log P, this is talking about lipophilicity, it should be of less than 5, and molar refractivity should be between 40 and 130. These have been some general rules that have been made. It doesn't mean that they have been so hard and fast and then it has to exactly 5 hydrogen bond on us. It's not exactly 10 hydrogen bond accept us kind of thing. It is more or less that, around these numbers, they do play. Of course, there may be some drugs which may not even fulfill any of these things, but still there are also been some drugs that can be taken up depending upon the necessity and the situation and demand. Okay, when you are talking about these drugs based on this Lipinski's rule of five, and if you qualify something as drug, then what is that? Based on the introduction I already mentioned, these drugs cannot be just been introduced into the market. Then we wanted to just find out the new processes that have been available now to make something else also as a drug rather than following the conventional methods. In that process, we have that one something called co-crystal. Then let's talk about it. What is a co-crystal and its evolution and its significance in pharmaceutics? This is what I just mentioned in the early introduction before getting into the slides. So let me just talk about it. What are these things? And when you talk about these things, how we come up to know about the concept of these co-crystals and its evolution and all that one, as you are all been familiar, now you see that one, we follow everything. We learned everything from nature. We learned everything from nature, either by observation, what I mean by observation, and then we make an experiment that is just like a photo. That what we just take a photograph that has been that gives a print, that's like experiment that we carry out in the laboratories. And sometimes we'll take the theoretical concepts into the picture. They are being just to address the systems in the quantitative manner, or we can also look at them as xerox of the photos, because the theory that whatever that we propose, there should be resemblance and then one-to-one relation between experiment, that is nothing but a photo. So that's why theoretical concepts that can be considered to be xerox of the photos. And then the other part of it is in the computation. So actually many people, they consider theoretical and computation and the one and the same, not truly speaking. The computation is different. It is been based on some totally different concepts than the theoretical system that we just do describe. The theory has been the one, the theoretical concept is the one that actually explains the experiment, whereas the computational one is itself create new experimental setup kind of thing. That's what we call quantification by graphic interfaces and simulation of experimental observations. That's what we call in silico, to support the experiments that is been in vitro to the biological experiment. That means what happens in the human body, that is vivo. So this computation is the one that brings a connectivity between the vivo and vitro, whereas the theoretical is the one that establishes the ground rules for the experimental observations. So this is something that people have to really make a distinction between various parameters that we just do follow. We do learn and then nurture and follow in the process before actually getting into the any learning. So let me just take this nature, how these observations, when I have to substantiate it, I'm sure you all agree that there is a famous personality that has been Leonardo da Vinci. If we just look at, he projected these things in a pictorial form. Just if you talk about it, helicopter. What I mean to say that one, Leonardo da Vinci, he said that nature is the source of all true knowledge, and how it can be shown pictorially from our own perspective. If you talk about our inventions, helicopter, okay, that you can see that it is been based on the butterfly, how it just makes it's flying. And similarly, you can just look at, compare that other figure on the right side, the parachutes that are been from some plants that are been almost having the same feature whenever it just spread into the air. And same thing that happens in our biological, in our universe also. If we just look at which is very cheap that we do consider, that is water. Water just will be represented as only H2O molecule in the form of H2O, simple molecular formula. But actually water has a very complex structure, that has been water cluster, where these water molecules get connected to each other through hydrogen, some force of attractions, and those have been called hydrogen bonds. Those hydrogen bonds bring these water molecules and attributes a lot of different properties. You are all been familiar in early education of intermediate or so. It was mentioned that water exists in ice, liquid, and then gas form because of this hydrogen bond feature differences in the hydrogen bonds. Whereas H2S, the same in the group, that has been always a gas because such hydrogen bonds cannot be formed in the molecules H2S, though oxygen and sulfur belong to the same group in the periodic table. Such a strength of hydrogen bond, such efficacy of the hydrogen bond, not only for small molecule like hydrogen, so small molecule like water, but you can also see that one in the complex molecule, which has been the responsible for our existence and human evolution, the DNA, that has been through this hydrogen bond between the duplex structure through the nucleobases in the form of vaccinic base pairs that is been between adenine and thymine or guanine and cytosine in the form of DNA or uracil in the form in the case of RNA. If we just look at the structure, the beautiful structure which has been accounted for our human existence, that has been not that has been with this hydro because of these hydrogen bonds, because of these hydrogen bonds that have been formed between the nucleobases, and also the proteins that we do next to that one to DNA important species in our biological system, biological processes. The proteins, they are also been excellently being coiled or sheets, beta sheets, and all these things are being through the hydrogen bonds. What is meant by these hydrogen bonds then? That means nature is able to just make a beautiful picture, combining these molecules with a weak forces that can bring together large molecules. It is almost like what we see in human evolution also, human interactions also. Each one of us being like a molecule, then we form a society with interactions, either brother, sister, father, mother, friend, or wife, and then the any other relations that we establish. These are all being intermolecular interactions, the relations that we do talk, and then that is what actually makes the cluster, and these clusters are the ones that they ultimately, depending upon the size, we just again make different clusters that can be like people from particular region, say Andhra, and the people from Odisha, one region. That means this is one cluster, and that is another cluster. This cluster and that cluster, they do interact. That is what happens in the case of the proteins, and then go on, and then India, and then West Europe, and they become much more global in the process. These are the interactions that what we notice that from the nature, we actually just can understand that the intermolecular interactions play a very significant role. And if those biological systems that are been following up these intermolecular interactions to stabilize and then form these global structures, the same thing that may also be possible if we can understand their properties effectively to develop novel systems that can be extended even to cure may develop different type of molecules that can go to the health sector in the form of the medicines. So for this one, what it is being these intermolecular interactions, then what chemistry, that is what it has been defined as beyond the molecules by Nobel laureate Jean-Marie Len. I don't know how many of you have been aware. In fact, he visited Bhubaneswar, at least to my knowledge, a couple of times. He visited and delivered lectures. I don't know exactly in any other place, but at least I know that he visited twice the KIT University. That's what I can recollect it. Surely he must have visited some other places also, perhaps your own institution as well. So what is meant by this chemistry beyond these molecules? Molecular precursors, that if you take two different units that are been green and red, and then karyotic covalent synthesis, and we can get the target molecule. That means red is been attached to the different green units, and then those molecules, they will be connected together, and those dashed lines that indicates that intermolecular interactions, and then that molecule now, which we call supermolecule, that can further extend in different directions in both 2-dimension and 3-dimension by self-assembly process, and then that can be considered supramolecular assembly. If we just translate that into the chemistry, you can see that there is a molecule that has been shown in pink. that has been trimesic acid having benzene moiety with three carboxylic acids. And if you just come down to that and you see that one carboxylic acid can be written as a dimer that through the interaction of OH group with the carbonyl group and forms a beautiful cyclic network. And that network, each carboxylic acid, when it extends, and if you see on my left side, then you can see that one that forms a very interesting chicken wire network. It forms a chicken wire, or you can say here the hexagonal network. Six molecules, they do interact and forms the hexagonal network, and that extends in the 2 dimension to form a chicken wire network. And also in 3 dimension, you can see that one that forms the really wide structure that has been shown on the extreme right side. So these are this is the way that simple molecules in the laboratory, they also can replicate whatever the complex global structures that have been found in the biological molecules. So further take it into the various domains, the various with various molecules, then we can understand that its extendability to in a large scale. That's what I would like to show you. Next example, that these intermolecular interactions not only for carboxylic acid, and if you just sorry, I think I have difficulty to show on a particular. Can you see the cursor? Are you people able to see the cursor?
No, sir.
Yeah, I tried to invoke the cursor, but it's not coming. But only annotator is here. I think probably this is the, yeah, some laser pointer is here. I have got it now.
Now it comes.
Fine. So now if you just see that one, this is with the carboxylic acid, but if you just look at the same thing that with the amide functional moiety, so then this is the kind of an interaction that you can actually see that amide, and of course here there will be hydrogen, one hydrogen that is being, just for the sake of simplicity, it is not shown. And it is purely carboxylic acid and this is purely amide. But if we just to see that one, if such a carboxylic acid can form like this and an amide can form like this one, if you bring the carboxylic acid and amide together, and I'm sure you can find interaction between a carboxylic acid and this amide. Please make a careful attention on this one. This has been of very great importance that when you go into the further slides. Of course, similarly, various functional moieties I have shown here. Let us focus on this one, and we'll just go into this one in the later cases. And also you can see that one here, carboxylic acid with various heteromolecules that you can see this one, okay? And similarly, if we just talk about the recent, very much recent one that is coming into the picture, that is the boronic acids. You may surprise what is meant by this one. A boronic acid is the one that has been having the boron connected to the two hydroxyl groups and some carbon moiety on this side. And these boronic acids, they form this kind of a network themselves. But if you just look at this network, this, if you ignore this hydrogen and this hydrogen, this is almost like a carboxylic group. And if you consider all the things together, then this will be like an amide. This will be like an amide moiety. So that means this has a different hybrid features of both carboxylic acid and amides. That means whatever that we may see here, you may able to see that kind of properties and with this boronic acid. So these are the some of the functional groups that have been identified based on the biological macromolecules that have been observed in the nature, in the biological systems, how they have been connected, and some of those have been some of these have been derived from those concepts to create new assemblies mimicking those biological systems. Now let me just take you further. So then if you just look at that, all these intermolecular interaction mediated compounds, intermolecular interactions mediated assemblies, they can just assemble in different ways. In what way that you can just look at it? Let's assume that this is a solid. This is a particularly one unit that has been coming. And then if we just, sorry, if we just look at, if these are been arranged randomly, okay, so then that's what we call it as amorphous material. And if we just take this one, this arrangement in this way, in a particular order, so then this can create one form. and then this can create, in a different way if you assemble them in a different way, and then you can, and such kind of different arrangements, they have been called polymorphs. And you can see that these are the polymorphs that what we see in the actually chocolate effect, which I am going to explain you little later on after a while. Okay. And then if I just take it further, sometimes these solids, they may not exactly connect to each other in the way that it's been shown for the form 1 or form 2. Something else is required. So that has been the something here, we just call that as solvent. Okay, and then in such a case, these things are being called solvates. That means you can see that the solid is being separated by solvent molecules. And sometimes the solid, when they're been connecting together, you may also find that sometimes they don't come directly next to each other unless some charge is been associated with them. That has been something that is of a different way the solids can be arranged. And you can see that one, suppose if the solid is been of different nature, coming compared to the next unit of the solid, that's what we get will be the co-crystal. That means the solid here is connecting with another plus. Here you can see that this one, this is another moiety. When they have been arranging like this one with a new moiety, here if you see that these are all been uniform, the same solid, but here it has been different units that are coming based on the, that's what I just mentioned. If you consider this as a carboxylic acid and this is an amide, together they come and form union, and they can interact and they form self-assembly and get the co-crystal. To make you more to impress you people more about it, instead of getting into any chemistry and physics of all these things, let me just give you a general example what is meant by these arrangements. All of you have been fully aware and noticed that one, when you have to construct a wall, what is the smallest unit that we can make use of to construct a wall? That is nothing but a brick. So consider this as a brick. When you have a brick, you can make a flat wall. Okay, a wall just in a uniform. So that is the way. That means there is a brick next to brick, next to brick, next to brick. All these can be arranged, and that is the one way. And you wanted to have a different with same brick, also you can create a different type of a wall, not necessarily square or rectangle, but you can also make some kind of a different shape. So that is nothing but form. This is the way that one other form. That's why we call it as form 2. Now if we just come to the solvate, what it is mean that when you are actually creating a wall, when you are creating a wall, just keeping a brick next to each other, it doesn't make anything. It doesn't give the strength. I'm sure all of you agree. So what you require to put? We need to put some cement between the bricks, some cement between the bricks. So that has been what we can consider this one as a solvent, because some material it has been put in between the kind of thing. So that's why this forms the brick. Now, if you just look at here, what is this one? This has been the charge. Okay, as you are all been might have noticed that when you are using bricks for the construction, actually if you use the dry brick, that will not give strength to the wall. Actually, one has to soak the brick in water and then put them next to each other. Then that will give the different strength and durability. That is nothing but charging the brick. Is the same thing here also. Charging, that is going to give you a different type of a strength, and then you can get the ionic solid kind of thing. So here, when you talk about the co-crystal, when you have the brick, we not necessarily that only one type of brick. You can always construct a wall with a different type of bricks depending upon what we the shape that we'd like to have it. So that is nothing but here co-crystal. When you talk about amorphous, if you are just notice that when bricks are being transported from a place to another place, we can actually unload them in different form. One is that one after the other brick that can be put down and make one of these shapes, or otherwise the entire truck can be unloaded just by pullover at one time, and you can see that time all the bricks will fall down and they will not be in shape. That is nothing but the amorphous. These things that we'll just try to readdress towards end of the presentation. What is the significance of these things for the in terms of the analytical techniques or analytical process? So with this information, I would like to just put back. We will go back to this concept that is that what we talked about the chocolates. If you just look at these chocolates, you all must have noticed that chocolates when we just taste it and you keep the chocolate for a longer duration and taste it, and surely you all agree with me. The taste of chocolate appears to be different. The reason being the chocolate, whatever that cocoa butter it is being used in that one, that changes its form, its internal structure, and form a different type of arrangement, and that's why the taste is going to be different. That means the polymorphs are going to be different taste. Similarly, the co-crystal, if it is a crystal, if a different more than one molecule, and if they assemble together, then there will be different type of a taste will go taste in sense. If you talk about property, then different properties will be coming up. So for example, that I would like to show here, the molecule that is being called cyanuric acid and melamine. If we just look at this as the cyanuric acid, which has been nothing but three cyclic ring with three nitrogens and three carbonyl groups that have been attached to the remaining carbon atoms. So this is the melamine, where we will have NH2. Of course, hydrogens are not shown here. So three amino groups. This is the melamine, and these interact together and form a solid network like this hexagonal that has been called that has been termed as a Rossetti network. And if you just look at this one, the cyanuric acid and then melamine, these two together form ultimately give a solid which has unusually high thermal property compared to either of these solids of cyanuric acid and melamine. And also, please do make a note here. The both of them are been interacting with a biological, with very weak intermolecular interphore interactions that are nothing but hydrogen bonds. But still, they have shown its ability to the great extent, altering the physical property drastically. We may wonder how it will be possible. Yes, when we talk about weak, then one bond is weak. But when you have the bonds, multiple bonds together, that strength is been tremendous. So that's why we can see that when the properties are going to be are being changed very much in this solid that is in the co-crystal. Let me now take you to the some other examples based on this one. I'll skip this one. So this I already introduced what meant by supramolecular. This kind of concept that has been termed as a supramolecular chemistry are brought out to us by Jean-Marie Len, and describing that as a chemistry beyond the molecule, as I mentioned already. So these are the other pioneers that contributed for the development of supramolecular chemistry, because any learning and then lecture that we've been giving, we always have to respect the forefathers, those who contributed their all the youthful time and dedicated time for the new science or the new features, new knowledge that can be imparted to the next generations. And mainly Professor McGlary, Professor Bill Jones, and Professor Javretko, Professor Yagi, and Professor Fujita. Unfortunately, I had a very strong association with all these people. We had long meetings in several occasions, and also discussions, fruitful discussions. Some of my students, they went and then worked with them as postdoctoral after the research that has been performed in after graduating from my laboratory in various places. Okay, now taken with a gratitude to these people, let me just take that what is the recent development of these co-crystals in the laboratory from our own research work carried out by my students. So if you just look at the citric acid, which has been very natural biomolecule that you can look at it, and having the three carboxylic acid groups and one hydroxy group, and when this has been crystallized with the to 1,2-bis-4 pyridyl ethene. You can see that the ethene, this is the double bond. And very interestingly, it forms three different crystal structures. That means three different morphologies. One is been noted as alpha, other one is beta, the other one is been gamma, based on the different compositions of these two reactants, that's citric acid and then the bipyridyl ethene. And of course, there is also another form that has been noted recently, that is delta form, which I'm not going to touch upon it at this moment. And now when I just take this one, you can see that one here. Okay. Here also with bipyridyl ethane, that you can see this one. You can see that one. This also forms different type of polymorphs. Okay, different morphs. And whereas when it is being replaced with the propane link, then it will be only one form that's going to be obtained. And similarly, with a different type of azo molecule, that means nitrogen. If you look at this, is the nitrogen position is para, and whereas it is been ortho positions, still you will get in these cases only one. Interesting part is that this ethylene one, that means ethene one, that means two double bonds with a double bond. This forms three polymorphs, and also with single bond also, then it forms the three forms. I'm just going to describe with this double bond one in a little bit, more in detail in this presentation. All complexes, different forms are all been characterized by single crystal X-ray diffraction and powder diffraction and differential scanning calorimetry and some spectroscopic methods. About which I'll give you some fundamental knowledge about the single crystal diffraction patterns towards end of my presentation. How these are being made? Just simply taken all the things in both the reactants in the flask and then observing them every day what is actually happening. And you can see that one in day one, those are the type of crystals that come, and then they do change next over a period of time into different shape. And in the day two, then another shape that comes, and you can see that one day three, another shape of it will come, that is in alpha form, and you can see that one as day four and day five. Why I'm showing this one, it is to the, especially to the students, to appreciate that research has been something that's not that we'll just make look at for something and it has been going to take time and then we leave it and then go away, and not that one. Actually, we have to, research has been embedded is embedded with the dedication. Okay, and that dedication come with the observation, with the continuous monitoring the systems. So this is what the student, Miss Samina did. Right now she is Dr. Samina. She did actually observing the flask of the reaction process, continuously monitoring every 1 hour, and she could finally found out these different forms, transforming the crystal's shape within the solution, in the flask, in the solution, or in the flask having these solutions. You will appreciate this more with this video that I'm going to show you here now. Is not showing up video? Yeah, you can see that one. It's a day two, and day three, that has been what it actually doing day four. I'm sure you can see that some of these crystals transforming. Can you see this one here? So here everything, it has been changed into the black actually. You can see this day two, this and day three, day two still is been day three. You can see that one, a lot of changes that have been happening here. And these are all been recorded by her using the in situ camera, and then observing almost every 1 hour. She used to record the things and then make notice these crystals, which have been formed because of these intermolecular interactions between the molecules that have been used in the development of these co-crystals. So if you just carry out the exercise and then if you look at the microscopic structure, you can see that in the case of the alpha form here, in the case of alpha form, this is the kind of interaction between the citric acid molecules, whereas in the beta form, citric acid is connected to two bipyridyl molecules, and in the gamma form, one citric acid molecule is connected to three bipyridyl molecules. Here it is only two bipyridyl molecules. Here there is no bipyridyl molecules. Okay, then what happens to the bipyridyl molecules? In this case, they insert between them. In this case, they already, you can see that one here also, you can already see that. And if you just analyze further to look at that, what is actually the three-dimensional structures, these molecules, they do arrange in the three-dimensional. This is the way that it is being. Okay. In this case, this is the way that it is. So what does it mean by that in three-dimensional structure that it has been coming like this? This is something like if you just take your suitcase where the clothes are being placed, and the way that each one can pack the clothes in the suitcases as it varies. Here also the molecules, they do vary in given box in the three-dimensional structure, three-dimensional arrangement differently, and these have been leading to the different forms. Where that means this is the internal structure. They all look like the same. Like once you just pack the suitcase with clothes and lock it, everything will look same. But if you open the different arrangement of the dresses inside, somebody might have put all pants together and all shirts together, but someone might have put pant and shirt together, and a set of pant and shirt, been packed, differently. So the internal arrangement. It is the same thing that we can see that internally these arrangements, which can be determined by X-ray diffraction process. Taken this one and having this, how one can measure these things, also characterize these things further by using the powder X-ray diffraction patterns. And you can see that one, these are been different patterns that you can see that this is the beta form, and this is the gamma form, and this is the alpha form. You can see that the different patterns that you are getting. And what is interesting thing that you can notice that these patterns, these forms, they do also change from one form to the another. These also can change one form to the another. And in this case, I show that one alpha form here, these things, they convert into gamma form. And in the next one, I'll show you the beta form converts into gamma form. And as a result, you can easily understand that gamma form is the stable. And if you just take the gamma form at any given temperature, that is the same gamma form that is going to remain. So this is the important fact that has been what actually plays a very significant role in this pharmaceutics. What is it, pharmaceutics? We all know that when they have been marketed, they will have the expiry date. The reason of that expiry date is nothing but the variation of the features of the molecular compositions with respect to the time. And the time is nothing but because of the environmental effect. The same thing here, you can see that alpha form, it is, expiry time at 150 degrees, after 150 degrees. Beta form, it has the expiry time after 100 degrees, 160 degrees, not even 100, 165 degrees. And then you can see that one in the gamma form, that has been totally no change. It remains as such without subjecting to any kind of conditions. Okay, without affected by any type of harsh conditions. Only thing that it simply melts after a certain temperature. So this has been the one that has been the polymorphic features that have been used to identify to bring a change in the actually shelf time of the drug molecules, which I will show you in the subsequent slides. So this similarly, apart from the citric acid, there is another important drug molecule that is the ethanamide, that is been, and also thiobarbituric acid, when they are being taken together, and this also forms different polymorphic structures that you can see here. This is the form one, where the molecules, they do associate with each other through the hydrogen bonds in this form of cyclic pattern. And here also it forms a cyclic pattern. But if you notice that one, this pattern here, these molecules are been coming together with these anchors, but whereas these molecules that are been longer distance, that you can see that. These are the two different type of patterns that we can see. And if you look at in the three-dimensional arrangement, these are the ones that means here they have been separated. Each block is been separated by the next molecule by certain distance, but here it is been separated by some ring formations in the process. So these are the ways that different arrangements that come in the internal structure analysis assessment, internal structure assessment when you do by the X-ray diffraction process. Now I'll get into the details how these things actually can be prepared, because that is most important for you people to understand, rather than actually the outcome of the final products that are being shown to you people so far. One of the things that solution method, how these co-crystals are being prepared by solution methods, evaporative co-crystallization, that's what we call, and then cooling crystallization and reaction crystallization. And the second process has been the grinding method, that has been simply taking the components together and then grind them in a mortar and pestle, or liquid assisted grinding method. That means take the components and add just 1 or 2 drops of the solvent and then grind them, and then automatically they do form the interaction between the reactants that are being placed in the mixture or in the mortar. And the other one is called anti-solvent method. I'm sure some of you must have been aware of it. Then when you are trying to crystallize, once is better the one that is not going to solubilize the compounds. If you use that one, then that works as anti-solvent. That is mainly based on the polarity difference between the solvents. And then slurry conversion method. That means now if we just take the molecule, those are not been actually solubilizing easily, then just sparingly soluble, and you just repeat make them by mixing, just forming. It is just like you add more solvent and then rotate it by just either in the rotaoperator, using rotaoperator, or maybe just simply using magnetic stirrer, and then decant the solution, and whatever the product that comes, then that will be the one that you can use it for the as the co-crystal. Of course, supercritical fluid techniques, that has been little advanced one. Let's not get to that. And then grinding methods, that has been here. You can see this one. So they are just simple mortar and pestle. You can take it up. Probably I missed out. I don't know whether I have in the other slide. You can also use another method called another technique, another equipment called ball mill grinding. Okay. Here, selection method, as I explained, solvent and some solid. It's not exactly crystal, but we generally consider, since it is even microcrystalline, and then stir them at room temperature, decant, and then dry them, and then you can actually use PXRD to characterize them. And then now coming to the our address present concern, what is meant by the pharmaceutical co-crystal. We talked so far with a mixed examples, but what is exactly the definition of a pharmaceutical co-crystal? A pharmaceutical co-crystal, in that one, there will be two different substances. In a co-crystal, there will be two different substances, but of which if one of them is been pharmaceutically applicable, that means like API, okay, API, active pharmaceutical ingredient is there, then that is called co-crystal, pharmaceutical co-crystal. It could be both the components can be APIs, but at least one of them should be API to be called something as a pharmaceutical co-crystal. So these co-crystals, they come through that, several types of interactions, including hydrogen bonding, that what I just mentioned. And the first co-crystal, which was actually prepared using the quinhedron, that was long back, 1844. So that you recollect it. I told you at the beginning, the era was different compared to the present scenario. In 1844 itself, it was made. It was very simple, but from that time onwards, if we just look at now, it may not be that easy to just claim the things compared to that what it was there in the 1844. And what is the objectives of making these pharmaceutical co-crystals? That if we talk about it, enhance solubility, enhance bioavailability, enhance stability. So these are the important factors generally people look at it when they want to make these pharmaceutical co-crystals. And then the basic principle that has been based in these co-crystals actually generate from some concept called crystal engineering. When it has been taken to API-based co-crystals is extensive and continuing to grow. And for this one, the main tool that has been helpful to develop new systems is Cambridge Structural Database. What is this one? It is a database like what we see that chemical abstracts and also the protein data bank, etc. Similarly, for small molecules, this has been the Cambridge structure database, which will have information about the three-dimensional crystal structures of organic and organometallic compounds. And these compound data, we can retrieve it. It's not only bibliographic, it also have the some very interesting features of the fragment search and then identifying the based on the properties and also the unit cell parameters, etc. And it can actually determine the hydrogen bonds predominance in between the specific molecules. One can actually make that also compute from the CSD analysis. And then you can also see that when what are the interactions between the functional groups? Sometimes they call them as supramolecular interactions. This is only for some general understanding. But what is important that about 86% of known co-crystals are hydrogen bonded. About 86% of crystals. So that means, all, as you see, as we already seen in the very beginning, at the beginning, the biological systems are all being connected by the hydrogen bond. Similarly, what are the co-crystals that are being generated using the methods and also some of the examples I know shown to you, they are being developed, formed through the hydrogen bonds. In fact, of course, they can be also formed these clusters using different type of intermolecular interactions like halogen bonds or chalcogen bonds, or the cluster to cluster metallic bonds, etc. And if you just look at what is the information that can be deduced from this CSD, mostly that carboxylic acid has been the predominant one. And then you can see that when alcohols that come into the picture. That is because mostly this carboxylic acid will go to the heteronitrogen atom very easily because it's an acid and base kind of a pair that will come into the picture. And then how to screen the, what is the screening method for the co-crystals? We can just talk about it. Screening methods to include hydrogen bond propensity. That means how good the functional groups. If you recollect in one of the early slides, I had shown so many hydrogen bond patterns. So that has been actually good for the screening purpose. What is that? Which specific type of that pattern is been could be evolved or could be formed by the functional groups. Under pKa rule, that means which pKa, at what pKa, what type of pKa molecules they do come together and they do form. And of course, this can be analyzed using the CSD as well. That is been the process that has been followed it up to screening of co-crystals. What does it? Those things have been elaborated here. Hydrogen bond propensity is the hydrogen bond formation between coformers, and the pKa rule, co-crystal formation can be predicted by the proton transfer between the acid and base. If it has been more prone to transfer, then we will get the ionic crystals, or if it has been less, then we will just simply get the co-crystal. And this has been one method that I told you. I'll just explain, explicitly talk about it. This is liquid assisted grinding. This is of great significance. Modification of solid state grinding by adding little amount of solvent. That's what I mentioned. What is this, real role of this solvent actually is happening? It actually serves as like a catalytic, as a catalyst, and we can talk about as a catalytic effect, and enhances the co-crystallization. So if we just talk about co-crystals in the other process, that is from solution, it takes to form the crystals and come out of the solution. But here, very quickly, within few minutes, we can actually get the co-crystals. And then can be used to prepare high purity crystals. Here in this case, when you have just been grinding them together, then you can get also in bulk. It also can get in bulk high purity crystals with a less time. Allows synthetic synthesis of polymer form of the co-crystals. Co-crystals, they can also be formed different polymers that we discussed already. This can be obtainable easily by liquid assisted grinding as well. Now, let me come into the realistic picture of what is this co-crystals in realm of the manufacturing industry. We all know that one, there are APIs that are been, say, for example, here we can see that Valsartan for the heart failure treatment. These are the drugs that are being used, they are developed for these particular diseases that to be treated, and manufacturers are being these companies that are doing. So any changes to be done this one, then you may have to like this carbamazepine. If you talk about it, this is a brand name that has been given this title, and it is anti-epileptic drug, and then they delivered by or manufactured by or marketed by Novartis. If we just to take these drugs, they have some action. But if they want to come with new drug, they want to come with a new drug, because these drugs have some problems. These drugs have some problems. They're not of surely of that 100% disease cure. So to change anything, we need to come with a new molecule. As I already mentioned at the beginning of my presentation, that takes enormous time, almost like 10 to 15 years. So let me just bring out a change in this. So that if you see that one, these co-crystals have been made, and also Prozac, the co-crystals have been made, and also the co-crystals of itraconazole and co-crystals. These are all been put in, made. Co-crystals of these drugs have been made and noted to be they have the different bioproperties, bioavailability, which I will exemplify you with a some example on the itraconazole and a couple of other examples. So before that, I would like so that why these are been the difficult process, regulatory considerations, how they could be negated by co-crystals, then by NDAs and NDAs claiming to contain a co-crystal form, applicant with the following data. What is that one? You just have to demonstrate that API and CoFA are present in that unit cell. That whatever the crystallographic information you have, both the molecules should present there. And also the co-formers are being have ionizable functional groups. That's of important in the process. And the dissolution and substantial dissociation, its co-crystal form occurs before reaching site of pharmacological activities. What it has been important for the regulatory to consider this crystal as a new drug without going to the any animal tests. Using that one, you can see that one, Bernawire succinic acid with a co-former, that means it is a co-crystal of with this succinic acid, the solubility problem was addressed. Solubility problem was addressed. And also bioavailability problem was addressed for aceclofenic, gallic acid and nicotinamide as a co-formers. And then relative stability, relative humidity of theophylline was addressed by co-crystallizing with azoxalic and malonic acid. And these are been, you can see that how these have been addressed. 92-fold of increase in the solubility for the darunavir, and whereas for the aceclofenic, this has been increased by at least 2 units. Okay, that availability, and also relative humidity that drastically increased, but of course percentage was not available here. And photostability was increased for the nitrofuranol with amino benzoic acid. Similarly, you can see that one so many, including simplest molecule that is been with the paracetamol, which has its own problem in the mechanical properties. So that was addressed by the using the solvent evaporation method, prepared and improved the mechanical property. So these are the ones addressed. Yeah, here that is what I would like to show the example here, itraconazole. And if we just consider this one, it is an antifungal drug that we can consider. And when you crystallize this one with various dicarboxylic acids, various dicarboxylic acid, dissolution profile is increased drastically. You can see that when the molecule, this has been the API and this is the dicarboxylic acid. They do connect like this one. And you can see that one dissolution profile, if you just take here, and then with the succinic acid, the dissolution profile was so high compared to that, whatever that it has been shown for the pure itraconazole, that is with the black glow, and whereas with the succinic acid, it reaches its highest dissolution rate, and as a result, this antifungal compound, it has been marketed, this using this co-crystal modification. Similarly, you can also say that on this BCS class 2 anti-inflammatory drug, okay, indomethacin. So this is also been marketed crystallizing with such with the saccharin, with the saccharin. Now I'm going to show you another example where the simplest molecule aspirin, which turned out to be different when the co-crystals have been formed in such a way that the old its precedent molecule, that is salicylic acid, how it was been wide how it lost its glory and it could be bring back into the picture. So we just noted that there's some benzoic acid and thalic acid, they do interact with adenine through the molecular recognition forming co-crystal. One of the students, then he just noted that aspirin as having the carboxylic acid, where it can be just crystallize with the adenine and then make corresponding bioavailability tests. Then what it was noted that when aspirin was co-crystallized with the adenine, instead of aspirin just getting into that, and it converts into salicylic acid and then form the co-crystals with with the adenine, and that salicylic acid it reacts with adenine depending upon the composition that is being taken into 3:1 ratio or 2:1 ratio or 1:1 hydrated form. Okay, when it what is interesting thing, when the bioavailability was tested, you can just see that one aspirin, its pH has been actually 2.9, whereas the salicylic acid, the pH was 2.7. You are all been familiar, I guess, that no, salicylic acid was initially the drug that used to be the molecule that is used as a drug for the problems related to what it is been being cured with aspirin later on. The reason for discontinuation of salicylic acid is because it develops lot of acidity in the body. So then if we just that's what actually made aspirin to be better with the 2.9 pH, that as a better drug rather than salicylic acid. But if we just look at here, 3.1 is the 3:1 ratio of salicylic acid and adenine, then it shows pH now 3.1. And 2:1 also shows 3.3. And 1:1 water in the form of hydrate, that has been 3.8. And also you can see that solubility that has been increased from initial salicylic acid solubility 2.1, but here it has been 5.680. So thus you can actually review to compare, to consider the abandoned drug back into the limelight through the co-crystal formations. And also here if you see that both salicylic acid and adenine, both of them are bio-friendly to make use this compound as a co-crystal. So this has been something that still we have been continuing to look into that, how to bring it into the bulk scale and also to attract the pharma industry to consider for its formulation further in the sector of business in the market. Okay. Another very important thing that this aspirin studies, it is in a way that the parallel experiments were done when we were doing that time also. There is a new polymorph aspirin was obtained, but at the same time in US also, Javaretko group, they just also came out with by this co-crystallization process, they could prepare the aspirin as a different polymorph. So both of us were at the same time carried out, but unfortunately we were not able to just put it up before them. So it has gone in their name. So that's because aspirin second form, this form was noted to be having more bioavailability, but that could not be synthesized until 2005. So this came out, but anyway, this did not get to the critically it is predicted to have the more bioproperties, but that was not the case when it is actually came into the picture for the realistic examples. In that way, we can feel that happy that we are not such a, we didn't go before Javaretko group, otherwise it would have been a big loss on everyone. So another instance is in the hydrochlorothiazide and pyrazinamide. These are the two drug molecules that are being taken and then form the drug protein. This has been where I told you both the molecules can be drug molecule APIs. So here you can see that physiochemical properties of the API of these either of the APIs that have been increased by using both the drug molecules, leading to the stability, manufacturability, solubility, dissolution rate, and absorption and bioavailability of these two independent drugs. And I'm going to end up with examples by simply showing this paracetamol with the phenazine example. There we can actually, as I already mentioned, paracetamol difficulty is a mechanical problem. Cannot make tablet that easily. But by crystallizing with phenazine, that could be easily tableting could be achieved. And this is in the process to just go into the commercial sector. That was done long is about 5 years back in the system. Of course, not from our group, this one. So another probably this is my last example, I guess. Okay, this has been very interesting part that actually led to the pharmaceutical industry. That is carbamazepine that actually has lot of issues about its solubility. But when it has been made with the succinic acid, the solubility problem was just been taken off with without any boundaries, and it has emerged as a very strong co-crystal that has already been marketed in the commercial sector. So the information that you get, based on this kind of topics that you will get, in the journals, that has been mostly crystal growth and design, organic research and process development, Kistingicom, Journal of Pharmaceutical Sciences, Journal of the American Chemical Society, Angwantech Chemistry, Chemical Communications, Advanced Materials, Advanced Drug Reviews, etc. These are the select ones, but there is also used literature that one can look at it. Then generally these molecules that have been made with the form of co-crystals, they will be characterized by X-ray diffraction. There are two types. One is been powder diffraction and another one is single crystal diffraction. The equipment, they look like this one. Okay. And this is single crystal diffractometer. Of course, these have been, both of them are powder diffractometer. One is from Philips and another one is from Bruker. So how this X-ray diffraction works and how that is going to be, these have been internal structure of the different parts of the equipment. How initially the information that will be seen, anything that has been coming like these parts kind of thing. So that means they have been good to process it. That's what it indicates that, your molecules have been well ordered. Okay. So then when you talk about this all been done by X-ray diffraction, that means subjecting the compound to the X-rays. But when you just look at, when you take this X-rays, there's a way that you get like these parts, then how to analyze these molecules? Okay, how to analyze the molecule from these parts? So initially it will be very cumbersome kind of thing, but ultimately you will get the beautiful structures, whatever I shown, all those things evolved, analyzing the dots that are being coming from here. As we all been familiar with X-ray images. When you take an X-ray for hand, though you've been, they all look like very scary kind of things here. But the scary images that you see, that can lead to that could be for this kind of human beings, sumos. Okay, they also will have the similar X-ray image, or it could be a very beautiful face that has been described by Satyajit Roy, film actor's brother, as a beautiful picture. And then also it could be a great scientist. We know that it has been Einstein of this century. So either of these people, their X-ray images are taken, it will be like this only, any part of it. Similarly, here also, the complex nature of these dots, if you analyze them, the way that we get these pictures, that we can also get the molecular pictures, very nicely. How this is going to happen, that one? So they're based on the Bragg's equation that is called n lambda is equal to 2d sine theta. The solids that are been, placed like parallel sheets, and then light falls on it and it diffracts, and then this equation that has been derived by Bragg, for which he was given awarded Nobel Prize. And how this works actually. So if we just, this is exactly like what we see objects under the microscope. In the microscope, if we have to see that an object, visible light will fall on the object, and then it forms, then it scatters, and then there will be objective lens that will combine together, and with eyepiece we can see the magnified picture of the object that we, whatever that we see. In the same way here also in X-rays, there will be X-rays object and X-rays will fall on that and they get scattered. But unfortunately in the X-rays, there is no lens available to focus them. So that's why these X-rays will be sent to the computer to do some Fourier transformations kind of thing. And then here a crystallographer will be there to understand, correlate these Fourier transformations to the realistic structure and bring a reasonably acceptable correlated structure. So that's what we can see on the screen, the molecule indirectly as an image rather than as a photographic picture that we see in the visible light. So this is the way that diffraction process that works to characterize the compounds. That's what I promised that I'll talk about the characterization technique at the end. I think this is the my end of the presentation, and I thank you all for your attention and the time given. And once again, I appreciate and warmly, gratefully acknowledge the invitation by management and Professor Pushni. Thank you once again.
Thank you, sir. The presentation was really informative, and we were very thankful to you for this very wonderful presentation. It has illuminated our brain and it has imbibed us with the various technologies that are being uh, implemented in the development of co-crystals. Thank you very much, sir, for this presentation.
Thank you.
Thank you. Thank you, sir.
Queries that people have, I can take.
Yes, yes, sir. You can share with us the presentation, sir. It was really informative for us, very educative. Thank you very much, sir, once again.
Okay.
Sir, before we proceed, there are some questions for you, sir.
Yeah.
So I would like to forward these questions, sir.
Yeah.
Just a minute.
Yeah.
Sir, there is a question from the side of the participants that could the co-crystals be a universal solution for solubility limited drugs, or do they just have molecular specific limitations?
Yeah, any development that any solution cannot be universal. Okay. Uh, Definitely, when you're talking about this co-crystal, the solubility, one of the problems to address, I had shown already, it is not just only for the solubility. There are bioavailability is another one. And then the tableting mechanical problems have been another type of things to be can be addressed. Okay. There are different problems, not only just to limited to the solubility. I hope that that's the question. Am I right?
Yes, sir.
Yeah.
Sir, another question from the participants. How might the pharmaceutical co-crystals integrate with personalized medicine in the future?
What meant by the personalized medicine in the in terms of it?
Sir, the question is that whether co-crystals can integrate or they can be used for the development of personalized or precision medicine. That is the question, sir.
Yeah, but then no. These co-crystals come into a picture where there is a difficulties associated with a particular drug. Okay, suppose if generally the drugs are not having any problems, we don't have any known problems associated with the drug utilization, etc. Okay, so then these co-crystals they may not come into any picture. So the personalized drugs, that the native drugs, they may not get replaced by this one.
Okay, sir. Sir, another question is that, sir, most of the phytomolecules, they are hindered by poor solubility. So, sir, can this formulations, that is the co-crystals, can address the bioavailability problem of the phytomolecules?
In principle, yes, but only thing that it will not. Unfortunately, what we cannot say that because some co-crystal that overcome with the solubility problem by taking the API with particular compound, and the same composition will work, the same coformer will work with other one also. We have to experiment it with different coformers and test it.
Okay,
And
okay, sir.
finally, we come out with appropriate coform. So in principle, yes.
Okay.
But only thing also to be taken into consideration, the co-farmer that we choose, that should also be bio friendly. That means it has to be, it must have been approved by this US Foods and Drugs Control Authority.
Okay. Thank you, sir.
Add something that is like, suppose say talk about benzene. We cannot take, co-crystal with benzene because
Yes,
that's
sir.
already been carcinogenic compound.
Yeah, it's carcinogenic, sir. Yeah. Thank you very much, sir, for addressing all the queries. So now I would like to take this opportunity to respectfully invite our esteemed Dean, sir, Dr. Amir Kumar Prushti, sir, to kindly come forward and present memento to Dr. Pedireddy as a token of our heartfelt appreciation and gratitude for this enlightening and thought-provoking talk. Sir, please.
Sorry. So thank you, sir.
Thank
And
you, sir.
once again, thank you, sir, for giving this provoking and insightful talk, and your simple language, and making us understanding how co-crystals can enhance the bioavailability and solubility, which is very important in some type of drugs which are not soluble and bioavailable. So thank you once again. And from the Raman Global University and Faculty of Pharmacy, please accept this token of appreciation, which I will send you later.
Thank you very much. Thank you.
Thank
It's
you. Thank
honor.
you, sir.
Yeah, actually, it would have been nice if I had come there, but I don't know there are limitations. So maybe sometime we'll visit. Okay.
Definitely, sir. Physically, we'll meet and we'll discuss about those things. And once again, I will invite you also physically so that you can input your knowledge to our students, so that will inspire more to the students.
I hope that I could convince the community, students, and also faculty, the
Sure, sir. Definitely. Yes, sir.
the
Yeah.
process of pharmaceutics actually.
Sir. Very nicely, very nicely. Sir.
Thank you. So shall I stop my sharing? Shall I close my?
You can
Sir.
share your screen, sir. Now I will request Ipsa to conclude the session.
Yeah.
Thank you, sir. Indeed, your valuable insights and contributions have greatly enriched today's session, sir. May I now request Ms. Priyanka Priyadarshini Patnaik to deliver the vote of thanks and officially close the session today. Priyanka, please.
Good evening, sir. Ladies and gentlemen, as we draw this enlightening session to a close, it is my privilege to deliver the vote of thanks on behalf of the organizing committee. First and foremost, I extend our heartfelt gratitude to Professor Dr. V.R. Peddi Reddy, Professor at the School of Basic Sciences, IIT Bhubaneswar, for his exceptional talk on the pharmaceutical crystals toward enhanced bioproperties of the drugs. His insights into the role of co-crystallization in enhancing drug properties have truly deepened our understanding and sparked meaningful dialogue. We are immensely grateful to our Dean Sir for his gracious presence and for his honoring our speaker with a memento of appreciation. I would also like to thank all our distinguished participants, faculty members, and attendees for their active engagement and thoughtful questions. We added great value to the today's session. A special thank to the technical and organizing teams working behind the scenes to ensure the smooth conduct of this webinar. With that, we officially conclude today's session. Thank you once again, and we look forward to welcoming you tomorrow for the future sections at the same time and at the same place. Have a wonderful day ahead.
Sir, namaskar, sir.
Okay, namaste. Thank you very much. Thank
We'll
you.
conclude, sir. Thank you.
Bye bye.
Yeah.
Recording stopped.